It's my pleasure now. I believe Dr. Lemke is on tonight, but we are so blessed to have Dr. Greg Lemke from the Salk Institute for Biological Studies in California. He's going to be talking about the role of MER. It's a multifunctional receptor tyrosine kinase in regulating vascular function in Alzheimer's disease. He is being awarded a $250,000 Roger Ackerman CART award. This is considered our top award for the 2020 to 2022 year. And we're just so pleased to have Dr. Lemke with us tonight to share what he plans to do with these funds. Dr. Lemke?
Yeah, our project, the title of the project is a little bit, we shortened it up a little bit. So we're talking about TAM receptor regulation of the vasculature in Alzheimer's disease. So I work at the Salk Institute. That thing you can see at the bottom of the screen is actually a picture of the institute. We're largely shut down right now. Only essential folks can come into the lab. We've had a little bit of opening this week. So we're actually starting to do some experiments again, and I'm hopeful that we'll get going on this project very, very soon. So our project deals with probably the most recognizable feature of Alzheimer's disease, which is the accumulation of plaques in the brain. These plaques, which in this picture are depicted as these sort of yellowish orange blobs, are aggregates of a small peptide called amyloid beta, or A beta. This peptide is in turn cleaved from a larger protein called the Alzheimer's precursor protein. And the aggregation of the peptide into large fibrils, which then get compacted into these large aggregates, is actually the hallmark feature, one of the two hallmark features of Alzheimer's disease. And so our project focuses on the vasculature, that is to say, the system of blood vessels in the brain. And why should we focus on the vasculature? Well, there's two reasons. One is your brain is your most highly vascularized organ in your body. So the density of blood vessels in the brain is higher than any other organ. And the other feature specifically that's important about Alzheimer's disease is that it's now known that most of this damaging yellow A beta peptide and A beta fibrils that are produced in the brain, during Alzheimer's disease, the way your brain actually handles with this problem is to get rid of most of the A beta protein, is to remove the A beta protein from the brain. And in order to do that, it has to cross from the brain over a set of cells that form something called the blood-brain barrier. And this is basically the vascular system of the brain. You have to get it out of the brain and into the bloodstream. So the blood-brain barrier, you can think of all the blood vessels in your brain as basically tubes. And outside of those tubes is the brain. They're a hard physical barrier. They're composed of several sets of cells we're going to be talking about in my presentation today. Endothelial cells, which are actually the cells that form the blood-brain barrier. You got to get the A beta out of the brain, across those endothelial cells into the bloodstream. And this is the main way the brain deals with Alzheimer's disease, because 85% of the A beta that's produced in the brain in Alzheimer's patients is actually transported across this barrier into the bloodstream and then out of the brain. That's the main way your brain handles it. And we've known for quite a while that the blood-brain barrier is really important. I just put one example of a recently published paper here from the Slokovich lab at USC here in California. You can see the title. It says that the blood-brain barrier breakdown is an early biomarker of human cognitive dysfunction. All right. Yesterday, in preparation for this talk, I did a Google search that scientists can do. I searched on our website called PubMed, and I queried blood-brain barrier and Alzheimer's disease. And the program turned back for me 3,176 papers. So we know the blood-brain barrier is important. A lot of people are very interested in it, but we don't know how the properties of the barrier, its barrier properties itself, are regulated. And this barrier prevents two things from happening. It prevents stuff from the blood that shouldn't be in the brain from getting into the brain, and it prevents stuff in the brain that shouldn't be in the blood from getting into the blood. So our lab studies a set of receptors, and this diagram I'm showing you here is sort of a complicated version of what these receptors look like. They're called TAM receptors. TAM comes from the three receptors that are in the family, tyro3, AXL, and MER. These receptors go through the membrane on cells. That's this TM domain here. This is the transmembrane domain. And they have a large part here, these things that are labeled IG1 and 2 and FN1 and 2, that sit outside the cell. And they're looking for signals. And there are two signals that I won't talk much about today, are ligands, which bind to the receptor and activate the receptor. These two proteins called Gas6 and Protein S. So you got three receptors here. Most importantly for what I'm going to talk about, these receptors have an enzyme, this red thing inside the cell, called a tyrosine kinase. And this enzyme is already being targeted by FDA-approved drugs. So we already have drugs in the clinic that either turn these receptors down or turn them up. So if we learn something about what they're doing in Alzheimer's disease, we already have approved drugs that we might be able to use, all right? And for the purposes of the talk today, the most important of these receptors by far is the receptor MER. So in the brain, MER is expressed by two main cell populations that are both important for Alzheimer's disease. One are the main immune sentinel cells of the brain, which are called microglia. So this red signal here is actually, you see, is MER expression in these microglial cells. But the other cells that express MER are these endothelial cells, these skinny little cells here, which line all the blood vessels in the brain. So this big black space that you see here is a capillary in the brain. That's where the blood would be flowing through. And lining every capillary and every blood vessel are these endothelial cells, these skinny little cells, which are marked by this gene called CD31, and they're also strongly MER positive, all right? So MER is in these endothelial cells, which form the walls of the blood vessel and which are one of the two key cells of the blood-brain barrier, right? So everybody knows Alzheimer's is a disease of aging in human beings, and we will do a little bit of work in human material, but we're mainly going to study a mouse model or a couple of different mouse models of Alzheimer's disease. So our main workhorse model is this guy here. He is a mouse that's called the APP-PS1 mouse. And this mouse is expressing human versions of that Alzheimer's precursor protein that gets cleaved to make A beta that causes Alzheimer's disease. And he's expressing a mutant form of APP and a mutant form of the protein that does the cleavage. And as a consequence, this mouse develops a lot of A beta in its brain. It eventually develops plaques, just like AD patients do, and it eventually gets very sick. So we're using this model, as this mouse is basically a model for what's going on in Alzheimer's disease. So you'll see in the subsequent slides, lots of things that are labeled APP-PS1, and those are these Alzheimer's model mice. Okay, our research is really predicated on, so we're doing a lot of work on MER in that other population of cells, the microglial cells, the immune cells, which are also very important in Alzheimer's disease in terms of what goes on in the disease. Actually, we're just about to submit an important paper on this. But the project that CART is funded was motivated by these observations. So first of all, if you look at the left, we noticed that the expression of MER itself, of this receptor, in those endothelial cells was reduced in mice that have Alzheimer's disease. So if we look at the level in this black bar, this is the level of expression in normal so-called wild-type mice. And we've actually quantitated this a few more times since this time. The level of MER expression in these Alzheimer's model mice, the APP-PS1, is reduced by about a factor of 2. So the levels of MER are down in these endothelial cells. They have less MER. And from other experiments that we've done in the lab, we already knew that MER was important for maintaining the tightness and the integrity of the blood-brain barrier in the brain. The thing on the right shows you that if we look at the expression of a whole bunch of genes, you don't have to know what these genes are, it's just a list of genes here on the right, that encode components of the basement membrane that stabilizes the blood-brain barrier. If we look at the expression of these genes in mice, in endothelial cells, so this BMEC here is brain microvascular endothelial cells. So these are the endothelial cells of the vasculature. If we look at their expression in a normal mouse versus a mouse that doesn't have any MER, a lot of expression is red and much less expression is blue. You can see that in 3 different mice we have that don't have any MER, that are knocked out for the MER gene, the expression of all of these genes that are in the blood-brain barrier is significantly reduced. So we have less of these components, these basement membrane components, that are required for integrity of the blood-brain barrier. So that's what we say down here. Endothelial cells without MER express much less of these proteins. This is actually not an RNA analysis, this is actually looking at protein expression by a procedure called mass spec. So they have less of these basement membrane proteins you need to assemble the blood-brain barrier. They also they express less of a couple of very, very interesting genes that are important for this transport process that I told you about before. So remember I said the main way the brain handles the A beta is to get rid of it. It transports it across the blood-brain barrier into the blood, and it goes out the blood system, and you eventually actually piss it out of your body, right? So these are looking now at gene expressions, again, in these endothelial cells, these brain microvascular endothelial cells, these BMECs, that are either normal, we call them wild type, WT here, or that don't have MER. And I want to point out 2 genes in particular, this gene ApoE and this gene MEOX4. So you may have heard about ApoE. ApoE is a famous gene in Alzheimer's disease. There are 3 different alleles for this gene in human beings. You can have a version that's called 2, 3, or 4. And if you have 2 copies of the 4 allele, you're much worse off than if you have the other alleles, especially if you have the 2 allele. Now, importantly, we know that the ApoE protein is expressed by these cells in the endothelial, expressed by the cells of the blood-brain barrier, including the endothelial cells and another cell immediately around it, the pericytes, and it's required for this transport process. Indirectly, it binds the A beta in the brain, it routes it through the endothelial cells and into the bloodstream. And what you can see here is that in these cells that don't have MER, again, the blue is less signal. They have much less of this ApoE protein than do normal wild-type BMEC cells. Oops, sorry, I went the wrong way. So ApoE is a very, very big thing. And both ApoE and this other gene, MEOX2, which is a different kind of protein, it's a transcription factor, they are required for this A beta efflux from the brain. And so without this efflux, and you're going to have less of these proteins in the MER knockout, A beta builds up in the brain more than it would normally if you were transporting it out, and Alzheimer's disease develops. And the final piece of data I want to show you, one thing that you see very, very commonly in Alzheimer's disease patients is something called cerebral amyloid angiopathy, or CAA. And this is the pileup of A beta and its proteins around blood vessels. This occurs in up to 90% of Alzheimer's patients, and it's thought to be a very important contributor to disease development in these patients. So here you're looking at brain sections here. The top 3 sections are either from an Alzheimer's disease mouse, normally, APPPS1, or the bottom 3 sections are the same APPPS1 mouse that doesn't have any MER, so the MER gene is called MERTK, and it also doesn't have any of the other axle protein. And then in the 3 panels, what you're looking at is the appearance of plaques, which is marked by this antibody called 6E10, it's this red signal here, or earlier, less well-organized oligomers and fibrils, which is this green signal here. And what you can see is there's a lot more of it. There are bigger plaques closer to the blood vessel, so this dotted line here marks the blood vessel in the section, and the lumen is the interior of the section. So what you see lumen is the inside of the blood vessel. You see this stuff is accumulating around these blood vessels much, much more dramatically in the Alzheimer's mouse, if you don't have the MER receptor, than it does in just the Alzheimer's mouse alone. So we have a model for what's going on here in the thing that this is really what we're going to test in our proposal. So in a normal Alzheimer's brain, so in the APPPS1 mouse, you are generating A beta normally in the brain, and this is going to cause a problem. And the main way that the mouse deals with this is to export this A beta across these pink cells and these gray cells and this basement membrane, which is the blood-brain barrier. It gets it into the blood, and the blood then, moves it out of the brain. That's what happens in a normal brain. If you don't have MER, you have all kinds of problems. So we know you have disruption of the blood-brain barrier. You actually have leakage of the blood-brain barrier. We know from data we already have in the lab that this barrier is leaky. And we know if you have a leaky blood-brain barrier, you're much more prone to develop Alzheimer's disease. So without MER, you have reduced A beta efflux. You have a weak blood-brain barrier, or BBB. You have leaky vessels, and you have A beta in and around these vessels. And you have, as a consequence of all of these things, worse Alzheimer's disease. So what are we going to do with the CART grant? Well, we're going to approach 5 questions. One is we're going to look at MER regulation in blood vessels, in the endothelial cells of the vasculature, and in parasites, in both AD sections from patients, from real human patients, and in mouse models. We're going to identify the full repertoire of all MER-regulated genes and proteins in endothelial cells, in ECs, during Alzheimer's disease and its mouse models. We're going to investigate the role of MER in the blood-brain barrier during AD and its mouse models. We're going to determine if MER truly does influence the rate and quantity of A beta transport across the blood-brain barrier and out of the brain. And finally, we're going to investigate the role of MER in cerebral amyloid angiopathy, this very, very important AD complication, this pileup of A beta around the blood vessels. Those are the things we're going to do with the money that you're providing us. We're really excited to get going on this. We're almost ready to get back in the lab. So the 2 people who are mainly going to do this are a postdoc in my lab, Kaisa Happanen, and a graduate student, Yutong Huang. And so on their behalf, I will simply say thank you very much. Turn it back over to you.
Dr. Lemke, that was a wonderful presentation. We're doing well on time. Are there any questions for Dr. Lemke?
Dr. Lemke, this is Norm Rogers.
I know.
We've had other doctors that have and I don't want to simplify it too much, but have made statements. One of them made a statement almost like everyone has Alzheimer's. Maybe that they don't have the disease, but they have the symptom. And the body, just like you're saying, it's almost like it gets rid of the garbage. And some bodies will, is that the MER? And then a second question I had was, with what you're studying, would that help us with other, like Parkinson's and things of that nature?
So those are 2 very good questions. So you're right. So the precursor protein that we're talking about, this APP protein that gets clipped at the end to make the A beta peptide, that's a normal protein. That's in everybody's brain. And this clipping is going on all the time at a certain rate. So it's really a question over time. This is why Alzheimer's disease is a disease of aging. If you just slightly accumulate you're eventually accumulating and accumulating. So as I said at the beginning, the most important way we have of dealing with this in the brain is not, for example, these immune cells that I told you about, the microglial cells that are inside the brain. They also eat up the A beta, and we're studying that process as well. But that seems to be a small component of things. The main issue, the main way, as I said, the brain handles it, 85% of that A beta that's produced is removed from the brain so that it can't aggregate into these problems. Now, that's why as people get older, their blood vessels tend to break down. So if you have problems, that's why people can have vascular dementia. You've probably heard about this as well. Yeah. So if you have problems with this transport process, you're not getting rid of all of the amount of A beta that you should, and so it stays in the brain, and that gives it a better choice to pile up. Now, you mentioned other diseases that have, where you have aggregates of proteins. You actually have amyloid protein. That's not the A beta amyloid you have in Alzheimer's, but you can have it in other diseases too. And there's also evidence that that amyloid has to be transported out of the brain as well. So it's possible what we learn about here in AD is going to be generally relevant. And I would again emphasize that the proteins we're talking about, like this MER protein, we already have drugs for it because it's implicated in cancer, right? So there are a lot of people being treated right now for cancers where the MER gene is overactivated with inhibitors of MER, right? And this actually might be a very bad thing if those patients are pre-Alzheimer's patients, right? So we also have other drugs that can increase the expression of MER, right? And that would be a good thing in this setting. So that's one of the things that we're particularly excited about this is this receptor and the TAM receptors in general are what are called druggable, right? There are people already making drugs. We've already got them for other purposes, and we might be able to repurpose them for Alzheimer's disease if we discover something useful from this research.
We also have the award of a plaque that we want to show you, Dr. Lemke. We've selected, our board has made the first award, the top award each year, going to the researcher who is identified as the first selectee of our research grants, will always be known as the Roger and Deanie Ackerman Award. You're the recipient of that award today, and this is a photograph of it, I think, that we have up.
So Dr. Lemke, if we were in Columbia, we would have a big check to hold up between us, and I will probably be bumping elbows next year, but we would shake your hand and present the check and this very nice plaque that honors Roger and Deanne Ackerman, the founder of the CART program, way back in 1996. So congratulations on your award this year. Just fascinating research, and we appreciate your wonderful presentation today. So with that, a check is being put in the mail, perhaps already, Bill, but it'll be there in a few days at best. So congratulations.
Yeah, I'm only holding the picture. I'm not holding the actual check. Johnny and Angus have those.
Righty, thank you both very, very much. I very much appreciate this. People in my lab appreciate this too. We made these discoveries only, maybe 6 months ago. You should appreciate how important this funding is, because we're at a stage in the analysis, even though I think it's very exciting, that if we tried to put in an NIH grant for this, they would still want more preliminary data. So we're going to do a lot of really good science, I think, with this award, but we're also going to try to leverage it into a much bigger award from the NIH. And that's something I think the CART program can really have some satisfaction from. So thank you very much.
Thank you, and I appreciate you mentioning that, Dr. Lemke. We've seen that in a number of our CART awardees that were able to go on and get millions of dollars in NIH grants as a result of this work. So we wish you the very best.
You nailed one of the tenets of our whole organization, Dr. Lemke, is that identifying great research and then helping those researchers get to that next level. We still consider ourselves the venture capital of the Alzheimer's research, and you made that point very eloquently. Thank you.